All right. Hey, everybody. Welcome back to another CRC meeting. My name is Michael Alden, and I'm a PhD student here at Clemson University working under Jim Faust. Today, I'll present some data on our experiments looking at growth analysis. We're hoping to provide a reliable, easy method for trialing genetics in-house with a focus on growing for extraction. Okay, so what is growth analysis? Well, growth analysis really just refers to any quantitative method of describing growth over time. And so a fairly simple, non-destructive method would be just measuring the length by width dimensions of the plant, right? But a destructive method, one that's more commonly used, is to measure the total dry weight over time. And so that would require you to harvest, a certain number of plants from your population and then get an idea of what the size of those plants or the actual mass of those plants are at specific intervals through the growing season. And so an example of that is on the right here from this data presented from Hunt 2017. On the x-axis, he has days in the year, and on the y-axis is mean dry weight per plant. And so we'd expect early on in the growing season that as the seeds germinate, they're going to be fairly small, and there won't be much progress until the plants actually start putting out some true leaves. And then we would expect, fairly rapid biomass accumulation. But once you get to the end of the growing season, and with corn, the end of the kernel filling, and once you get to the physiological maturity, then you'd expect that there won't be any change anymore. And this is a pretty good indicator for when to harvest, right? And so the same idea can be applied to cannabis, right? As the growing season progresses or the crop cycle progresses, the flowers are not going to grow indefinitely. And so there'll come a point where the flowers stop growing. And similarly, we're interested in how are the cannabinoids changing over time? Are they just continuing to accumulate, or is there a point where they start to decrease? And so with this experiment, we measured primarily the inflorescence mass and the cannabinoids. And terpenes would also be something very relevant and interesting to measure. But for these experiments, we didn't measure terpenes. Okay, and so when we're trying to identify or predict our optimal harvest time, we of course need to think about what product are we going after. And so there's really two main classes of products. Number one being the intact dried inflorescences, or our flower buds. And these really have an emphasis on qualitative characteristics, right? The compactness, the aroma, the color, these things that seem to add a lot of value to the product with, maybe we don't necessarily understand how these things are necessarily correlate with the smoking quality of the product, but one that just the visual appearance will add more value to it. Now, identifying the optimal harvest time for flower buds is really complex because of how subjective the interpretation of the final product is, right? But if we compare that to our second type of product, extracts and concentrated products, we're specifically interested in maximizing the yield of our cannabinoids, right? We don't care what the flowers look like because the customer will never see the flower. And so the optimal harvest time is really just a simple calculation of how much flower mass do you have and what is the percent dry weight of the cannabinoid of interest. So right now, there's really two main methods that are used for determining when plants get harvested. Number one is that just there's a fixed number of weeks that the plants will be grown for. And then, let's just say it's an 8-week crop. Well, on day 56, they go into their facility and chop all the plants down. That one's just kind of a very cut and dry approach, whereas the other approach is evaluating kind of the trichome color fraction on the terminal flowers. And so we know that the trichomes will start off as these clear white in color, and then as time goes on, they will change a little bit more milky, then eventually get a little more amber and orange, and then finally they get brown. And the brown coloring is a little bit less desirable, and so usually growers are trying to hit some sweet spot where 30 to 50% of the trichomes on the inflorescences are in that milky to amber region. And so this is some data from an experiment that came out this year, and they're modeling or showing the probability of finding these different trichome colors as the flowering time progresses, right? So this would be 20 days after starting to flower, up to about 70. And so they're showing, the amount of these white or these clear trichomes will decrease over time. The milky ones will increase, and these brown ones will also increase as flowering time progresses. So, this is interesting. I think it probably does correlate pretty well with the actual maturation and the overall progression during flowering. But we're making the argument here that when we're growing for extraction, we don't care about this at all. It might be useful and it might correlate pretty well, but instead there's a much easier way for us to determine when plants should be harvested. So for our materials and methods, again, we're focusing on a mathematical approach to identify optimal harvest time without ever having to look at the trichome color. So similar to what we saw earlier with corn, we grew a little population like this in the greenhouse, and we would randomly select plants from this for destructive harvest. Inflorescence and vegetative mass were separated, dried for 3 weeks. The weights were recorded, and then inflorescence samples were prepared for cannabinoid analysis. Plants were provided a continuous liquid feed for the entire experiment, right? We didn't want to restrict growth at all, so no flushing was done. And so as you can see in this table, we put plants under 12-hour photoperiods for up to 10 weeks. And so we've done this experiment kind of in 3 different ways, once with THM Jack and twice with Southern OG. Now we're just going to focus on the data that we have with Southern OG for this today. And so with OG, we finished plants either with 16 or 8 shoots per plant, which you can see up here. And so they got the same duration of the vegetative period before starting flowering. The only difference is that the 16 shoots per plant treatment was pinched 1 additional time after starting short days. And I'll show you guys what I mean on the next slide. And so again, starting on week 4, we would go in, harvest 5 random plants, and then go ahead and just randomize the remaining plants that were still in the greenhouse. And so for OG with 16 shoots, we harvested plants every week from week 4 through 8. And then on this experiment with 8 shoots per plant, we went from week 4 up to week 10. And each of these experiments were replicated twice. So just want to run through the Cancedia pinching strategy with you guys again. We start by pinching these plants at the start of when we transplant them. This is 2 weeks after we take cuttings. They're usually rooted right about day 14. We transplant, pinch back to 2 nodes. We allow those 2 lateral shoots to develop, and then we pinch back again. And so each time that we pinch these plants, we're always leaving 2 nodes behind. When we get to this 3rd pinch, this would be 4 weeks after we took those cuttings initially. This would be pinch number 3, and we'd start short days here, and then this 4th pinch would happen 1 week after starting short days. So for our 16 shoots per plant treatments, they were pinched these 4 times, the final pinch being 1 week after starting short days. Whereas with the experiments with OG and 8 shoots, it would have stopped being pinched here, right? So it would have been pinched for the last time at the start of short days, and that's it. All right, so on the x-axis here, we have weeks of 12-hour photoperiods. On this left figure is the 16 shoots per plant experiments, and on this right figure is the 8 shoots per plant experiments. So the 16 shoots per plant, these went from weeks 4 to week 8, whereas on this right figure, the 8 shoot per plant, these went from week 4 to week 10. On the y-axis, we have dry weight, so this is in grams per plant, and we have vegetative, inflorescence, and total being the combination of inflorescence and vegetative. And so we see for both these experiments that from week 4 onwards, that the vegetative weight is flat, right? There's really no changes here. So all of the growth is occurring, beyond week 4 is occurring in these inflorescences. And so we see with the 16 shoots per plant experiment that each week, each additional week under short days, going from week 4 to week 8, we're getting, nice linear increases, these large increases every week. Now, if we look at the right figure, going from the 16 shoots per plant now to the 8 shoots per plant, we see a pretty similar trend here going from week 4 up to week 8. You're getting, nice large increases every week, but this starts to taper off for sure as we start to get to week 9, 10. And this is kind of what we'd expect, right? Because we know these plants aren't just going to continue linear growth indefinitely, that they will eventually slow down. So this is the same setup as the last figure where we have weeks of 12-hour photoperiods on our X. We have 16 shoots per plant here on the left, 8 shoots per plant on the right. But on our y-axis, we're just looking at inflorescence dry weight. So we're just zooming in on this. And so what we're looking at here then are these 2 lines are the 2 replicates for each of these experiments, right? And so, we see with this 16 shoots per plant figure that, the increases each week are pretty consistent and they're pretty similar between the 2 replicates. They look pretty much identical as they go through. And so that's why on the last figure, the error bars are, fairly small, fairly consistent. Whereas if we saw with this right figure, with the 8 shoots per plant, the error bars were much bigger. And this is the reason why. With 1 rep, we had, a pretty similar trend as we see at the 16 shoots per plant. But compare that to the 2nd rep of the experiment, which was much larger and a lot more chaotic, right? And so I think that's something that's more of a symptom of having fewer shoots, is that you're just getting much longer shoots, these inflorescence shoots that are developing, a little more variable between these experiments. Whereas when you pinch to 16, the growth is going to be a lot more uniform and consistent. So just kind of focusing on this difference that we're seeing with comparing the 8 and 16 shoots per plant. We're always advocating for additional pinches to at least get to 16 shoots per plant. And so that looks more something like this. This is one of our 16 shoot per plant treatments here. And so, when these additional pinches are forcing more of the flower mass up to the top of the canopy, rather than having flower mass that's down here is more shaded and not getting as uniform light delivered to the actual flowers. And so this is a study here. This data is from Danziger and Bernstein. And so what they're showing is they're measuring THC on this left figure and then CBD as a percent dry weight in this right figure. And so they took samples and evaluating the amount of THC in the actual flowers and then the inflorescence leaves, or these bracts surrounding the flowers. And so they did this at the bottom, center, and top of the plant. And so this is one of our plants from a few years ago when we were growing unpinched plants. And so this is kind of what it would be generally like the bottom flowers, the center flowers, and these terminal flowers. And so, just focusing on the THC figure here, at the top of the flower, like you'd expect, or the top of the plant, you're going to have the highest amount of THC. And as you get lower into the plant, that you're going to have less and less THC in these flowers. Now, what's interesting is that if you also compare that with the inflorescence leaves, the inflorescence leaves at the top of the plant have just as much THC in them as flowers at the bottom and center positions, that's really interesting. And so, it's kind of a good thing to think about of why these extra pinches are so important. So that makes, you're trying to eliminate these flowers that are in the bottom and center part of the canopy and get as many as you can in the top part. All right, so getting back to the data from this experiment, this is the output that we got from the cannabinoid analyses from the flower samples that we sent off. And so we have weeks of 12-hour photoperiods on our x-axis going from 4 to 10. And on our y-axis, we have CBD percent, so this is percent dry weight, THC percent, and then total percent. So this would have been the total amount of cannabinoids that were measured. And so the blue line is our 16 shoots per plant, and the red line is with our 8 shoots per plants. And so, the trends here are pretty similar for all three, CBD, THC in total. So I'll just focus on CBD percent here. And so between the two experiments too, the trends are almost identical. They're a little bit different, but similar in how they accumulate over time. And so going from week 4 to 5 and 5 to 6, we see pretty large increases. But then as we get to week 6, going to week 7 to week 8, this is pretty flat for both of these, the 8 and 16 shoots per plant. And they're actually almost right on top of each other for week 8. And then we do see this start to go down, and so there's pretty obviously that there's a sweet spot here. Where between week 6 to 7, where the CBD percent is the highest, and then as you go later into the experiment, as you go later into flowering, well, this percentage starts to drop off a little bit. So I wanted to put these figures side by side here, our inflorescence dry weight up on this top figure and CBD percent on this bottom figure. I thought it was pretty interesting seeing how the changes that happen each week, how they're different from each other. They are similar. Let's say from weeks 4 to 6, they're both increasing, right? Inflorescence dry weight and CBD percent are increasing in this region, right? But starting at week 7, this starts flattening off. This isn't going up any higher, and is actually starting to come down as time goes on up till about week 10. And if you compare that to the inflorescence dry weight, going from week 6, 7, 8, 9, 10, it is going up every single week. It is starting to taper off, starting to slow down a little bit, but it's showing kind of the difference that the plant is might be accumulating more and more flower mass every week, but that's not necessarily true about the synthesis of these individual cannabinoids. So here we're looking at the CBD plotted against the THC percent dry weight from our cannabinoid samples. And so the different colors here are representing the different weeks of flowering that we took the samples. And so it is pretty interesting that throughout the flowering cycle that the relationship between CBD to THC stays fairly fixed, right? This is a slope of 19.7. So pretty much at any time during flowering, the relationship between CBD and THC is about a 20-to-1 ratio. And so for regulatory purposes, you can really distinguish, hemp from marijuana genetics at any time, and the inverse as well. All right, so let's take a look at these yield calculations for our different cannabinoids here. So up here we have CBD yield, this is THC yield, and then total yield, so all cannabinoids that were present in the samples. And then x-axis, of course, we still have the weeks of 12-hour photoperiods. Same setup as before, where we have the 16 shoots per plant on the left side here and 8 shoots per plant on the right side. And then these two lines are just rep 1 and 2. And so with the 16 shoots per plant, just these three are all the same, so I'm just going to focus here on CBD. Each week that you go further into flowering, you get a significant increase in the amount of CBD yield that you're pulling off from your plants, and so it makes sense to continue going at least until week 8. And there really wouldn't be any reason to stop, because you haven't hit the point of diminishing returns, right? Each week of flowering, you are getting significant or large returns on your time that you're spending in flower. Compare that to the 8 shoots per plant, where it's not as clear here, and definitely a little inconsistent between these reps. So for rep 1, just with the CBD here, as we increase our flowering time all the way up to week 9, we are getting large increases. So it makes sense to go to week 9 here. And then week 10 isn't any different. So there won't be any reason to go to week 10. You would harvest right here and get your next cycle going. Versus this rep 2, where we had large increases going up to week 7, but then week 7 isn't any different from any of these later weeks. From 7 to 10, this is all the same. And so there really wouldn't be any reason to go past week 7, because you're not getting any more benefit from going further into flowering, and you really always want to be on this part of the growth curve, right? You always want to be adding significant mass, or accumulating more cannabinoids each week, and you don't want to be on this spectrum where you're these weeks where you're not actually accumulating anything. They're kind of going stagnant. All right, and so far we've really been thinking about our CBD yield or cannabinoid yield on a per plant basis, almost with the idea that it's just the singular crop that you're growing, and you're just trying to identify where am I not really getting any benefit from going any later into flowering, right? But now we wanted to take a look about thinking about this more on an annual basis, right? Because most of you guys are not just growing one crop per year; you guys are growing multiple crops per year, and so that's something to consider here when we think about how many times are we harvesting per year, and so that's what we're taking into account here with our annual CBD yield here on our y-axis. So taking our the grams per plant that we're harvesting from these plants, and then also considering how many harvests that you are doing per year. And so for, between this left figure, this is our 16 shoots per plant, and then we have our 8 shoots per plant here on the right, but we'll just focus on this one first. As we go from weeks 4 to 5 to 6, we are seeing large increases this whole time. Then as we go from 6 to 7 for rep 1, really not much benefit here, but going from week 7 to 8, a sizable increase here, right? And so that would make sense to go from week 7 to 8. On rep 2, we got a decent increase going from week 6 to 7, but not really much increase going from week 7 to 8. And so, of course, it would be very helpful to have maybe 1 or 2 more reps of this. If you're in a sole source environment, you might just decide to hit it right in the middle and just call it there and just be done with those valuations. But maybe if you're in a greenhouse, you'd maybe want to try this at a couple different times of the year and a couple more reps, maybe expand your sample numbers. But this gets you pretty close to trying to identify where your optimal harvest time is, being somewhere really between weeks 7 to 8, without ever having to look at the trichomes. Right? We never had to look at trichomes, stigmas, not evaluating the colors, not doing really any of this kind of subjective or maybe something that's a little more open to interpretation, just taking a purely mathematical approach here. Now, between the harvest per year here, you're really only looking at the difference of one harvest going from 7 to 8 weeks of flowering. And so maybe that's not as big of a deal, but compare that to the right figure here. Now we'll just focus on rep 1 here. As we go from weeks 4 to 5 to 6, we're increasing our annual yield pretty sizably. But across from weeks 6 to 10, there's really no difference here, right? These are all the same. And so when you look at the number of harvests you do per year, if you're harvesting at week 6, you're doing 8.7 harvests per year versus harvesting at week 10, you're only doing 5.2. And so, just thinking about, let's say the labor costs here, well, why would I want to do 3.5 more harvests per year if I'm not getting any more CBD on a yearly basis, right? And so that, that's a very good argument for that. And especially too, that as you do more crop cycles per year, that's going to put a greater demand on your, let's say stock plant productivity and your cutting propagation, right? You're going to need more cuttings, you're going to need more pots, you're going to need more substrate. So those are things to think about. But then again, if we also think about that, as we saw before, that each week that we go into flowering, that we are getting sizable increases in our inflorescence dry weight. Well, if we harvest at week 6, right? We have much smaller flower mass. And if we also think about with the CBD percentage, for example, it was really at its peak about week 6 to 7, but then started to come down, right? So you have smaller amounts of flower at week 6 and at the highest concentration, right in this time, week 6 to 7. Versus where you get out to week 10, yeah, you're adding more mass, but now you're decreasing your CBD percentage, right? And so really by going out later into flowering, you are really diluting your flower mass, right? And so that ends up making it so that you have to have more space for drying, right? You actually have more physical mass that you have to deal with. And that's of course going to put more demand on the extraction equipment, all the materials for that. And then also considering too that, maybe there's some differences between planting density here, because these calculations are assuming that you just have the same planting density. But if you're harvesting at week 6, you might actually be able to fit, a few more plants per meter square versus week 10. These branches get, really tall and heavy, and, it might be a little more challenging to, have a higher planting density. And so, these are some of the things to be thinking about as you try to identify your optimal harvest time. And then, of course, we didn't measure anything on terpenes, but, if you're also extracting, cannabis-derived terpenes from this, this would be also something to measure and take a look at, taking a purely mathematical approach here without having to do any of this subjective, trying to decide the color fraction of the, trichomes and things like that. All right, so just to wrap things up here, we've been thinking about trying to have a more standardized approach to determining when to harvest plants, right? Let's take the subjective parts out of this. Let's get something that's just a little bit more uniform, something that can be applied to all sorts of genetics, and make it just a more simple approach to determining harvest. And so we're defining optimal harvest time for extraction. We understand that it's a way more complex when we're thinking about smokable product, but when we're talking just about growing cannabis for the purpose of extraction, we can define our optimal harvest time as the minimum number of weeks in flowering required to obtain a near maximum yield. Our second point being that we saw pretty drastically that pinching strategies can affect the consistency of cannabinoid yield per crop cycle. Our additional pinches resulted in more uniform growth, and I think this makes sense. With the additional pinches, we're pushing more of that flower mass to the top of the canopy. We're getting more photon capture directly at those inflorescences, and then we're not having this variable light levels throughout the canopy. We have all our flowers right at the top, all exposed to the lamps or exposed to the light source, and we should expect more uniform growth that way. And our third point, that CBD and THC percent plateau much earlier than the growth of inflorescences. This was definitely a little surprising, but maybe also to be expected. But seeing that the inflorescences just continued growing even up to week 10 was really interesting, and that it was starting to slow down after week 10. I definitely thought by maybe week 7, week 8 that we really weren't adding any more mass by then. It was just the flowers were just kind of maturing at that point. But we saw that the flowers will continue to grow, but the CBD and THC percent, and I'm sure many of the other cannabinoids, do begin to decrease. And so that's all I have for you guys. Thanks so much.